Welcome back to my channel Knowledge Chronicle.
Today, we're starting an exciting two-part journey into a very important and fascinating topic from your Class 12 Biology syllabus: pre-fertilization structures and events. Part 1. Make sure you watch till the end, because today's session will clear all your concepts with examples, tricks, and fun explanations. So without any delay, let's dive right into the microscopic world of flowers and reproduction. Much before a flower even appears on a plant, the plant has already made an important internal decision. It has decided to flower. How? Thanks to complex hormonal signals and structural changes inside the plant. At this stage, the plant begins developing tiny structures called floral primordia, which are the starting points of flower formation. These primordia eventually give rise to inflorescences, the bunches or groups that bear floral buds, which later bloom into flowers. And within these flowers, something magical happens: the development of reproductive structures, the androecium and the gynoecium. The androecium is the male reproductive part and consists of stamens. The gynoecium is the female reproductive part, made up of pistils. Both play a crucial role in the journey leading up to fertilization. Let's zoom in further. The stamen is the male reproductive organ. It has two main parts. First, the filament, a long, slender stalk that holds the anther. Second, the anther, the terminal structure, usually bilobed, where pollen is produced. You'll notice the filament connects to the flower's thalamus, or petal. Interestingly, stamens can vary greatly between species. Some are long, some are short, some curved, some straight. Nature is full of variety. Now, if we look at a typical angiosperm anther, it is bilobed, having two lobes. Dithecous, each lobe has two chambers, or theca. Running between the theca is often a visible longitudinal groove. But what lies inside the anther? Let's take a transverse section, TS, and see. An anther is a tetragonal, four-sided structure with 4 microsporangia, 2 in each lobe. These microsporangia later develop into pollen sacs, filled with pollen grains. In TS view, a microsporangium shows 4 important wall layers. First, the epidermis, the outermost protective layer. Second, endothecium, just beneath epidermis, important for anther dehiscence or opening. Third, middle layers, 1 to 3 thin layers, which later degenerate. And fourth, tapetum, the innermost layer, nourishing the developing pollen. Now, the tapetum is fascinating. It's rich in cytoplasm and often has 2 or more nuclei. Can you imagine? Because it needs to support thousands of growing pollen grains. At the center of the microsporangium is the sporogenous tissue, dense groups of cells that will form microspores. So microsporogenesis is basically the process where the sporogenous cells go through meiosis to form microspore tetrads. Each sporogenous cell actually becomes a pollen mother cell, or PMC for short. The PMC then undergoes meiosis, which is a reduction division, and this results in 4 haploid microspores. As the anther matures and dries out, these microspores separate and turn into individual pollen grains, all set to travel and help with fertilization. And get this, each anther can produce thousands of pollen grains. Isn't that just a marvel of nature? Now, let's chat about these tiny but super powerful structures, the pollen grains. They're usually spherical in shape, and their size is about 25 to 50 micrometers in diameter. Each one is surrounded by a 2-layered wall. The exine, or outer wall, is made of sporopollenin, which is actually the most resistant biological substance known. Then there's the intine, the inner wall, which is made of cellulose and pectin. The exine even has germ pores. These are thin spots without sporopollenin, and that's where the pollen tube will eventually grow. You might wonder, why such a tough outer layer? Well, it's there to protect the male gametes from harsh environments. Now, inside the mature pollen grain, there's a vegetative cell. It's larger, packed with nutrients, and has an irregular nucleus. Then there's the generative cell. It's smaller, floats inside the vegetative cell, and is spindle-shaped. In most angiosperms, pollen is shed at the 2-celled stage. But in some, the generative cell divides inside the pollen, and it's shed at the 3-celled stage. Pollen grains are actually nutrient-rich. Believe it or not, some athletes even take pollen-based supplements. But on the flip side, allergies can be a real problem. Some pollen, like from parthenium or carrot grass, can cause severe asthma and bronchitis. Pollen viability, by the way, really varies. In rice and wheat, pollen is viable for only about 30 minutes. But in species like mango, it can stay viable for months. Here's a cool fact. We can actually preserve pollen for years at -196 degrees Celsius in liquid nitrogen, which helps us build pollen banks for crop improvement. Alright, now shifting focus to the female reproductive part, the gynoecium. So, a pistil has 3 parts. First, the stigma, which is this sticky platform where pollen lands. Second, the style, which is a tube-like structure leading down. And third, the ovary, that's the basal part, containing ovules. Now, let's talk about types of gynoecium. Monocarpellary means there's a single pistil, like in pea. Multicarpellary means there are multiple pistils. If the pistils are fused together, that's called syncarpous, for example, tomato. If they're free, that's apocarpous, like in lotus. Inside the ovary, you'll find the placenta, from which arise the ovules or megasporangia. Just to give you some examples. In wheat, there's 1 ovule per ovary, but in watermelon, there are many ovules per ovary. Alright, let's zoom into the structure of a typical ovule. So, first up, there's the funicle, which is basically the stalk that attaches the ovule to the placenta. Then, we've got the hilum, that's the junction between the funicle and the main body of the ovule. Next, there are the integuments, which are protective layers, but they leave a tiny gap called the micropyle. Inside those integuments, you'll find the nucellus, a tissue that's rich in nutrients. And right within the nucellus, the embryo sac develops. That's the female gametophyte. Oh, and at the opposite end of the micropyle, there's the chalaza, which is basically the base of the ovule. Now, inside the nucellus, one cell actually differentiates as the megaspore mother cell, or MMC. The MMC then undergoes meiosis, which produces 4 haploid megaspores. But in most plants, only one of these megaspores survives, while the other 3 just degenerate. The surviving megaspore gives rise to the female gametophyte, or embryo sac. And by the way, this whole process is called monosporic development. So, let's talk about the formation of the embryo sac. The functional megaspore goes through 3 rounds of free nuclear mitotic divisions. First, there's the 2 nuclei stage, then the 4 nuclei stage, and finally, the 8 nuclei stage. In the end, you get 7 cells and 8 nuclei arranged in a specific way. 3 cells at the micropyle, which make up the egg apparatus. That's 1 egg cell plus 2 synergids. Then, there are 3 cells at the chalaza, called antipodals. And finally, there's 1 large central cell that contains 2 polar nuclei. Special structures, like the filiform apparatus in the synergids, actually help guide the pollen tube to the egg. And that's it for today's super detailed exploration of pre-fertilization structures and events. Part 1. We've covered every corner, from the flowering decision all the way to the magical structures of pollen and ovule. But hey, hold on. The story doesn't end here. Tomorrow we'll continue this fascinating journey with Part 2, where we'll dive into pollination in the same detailed and engaging style. So, stay tuned, because tomorrow's session is going to be even more exciting and honestly mind-blowing. Before you go, don't forget, your 1 like, share and subscribe will help us create more valuable content for you. This is Knowledge Chronicle, signing off. See you tomorrow with Part 2.